I will introduce the
Hello.
topic. Hello.
Good evening, sir. This is Naveen. I am
Yes.
working as a study scientist in in vitro toxicology, and I did a MS microbiology. That was my interest. I am participating in this conference, sir.
Okay. Very good. So I will suggest you, Naveen, keep in touch with the Bipran sir. His
Ah,
contact number is given.
yeah, sure, sir. Yeah,
Okay.
yeah, sure, sir. Sure.
So biopesticide is a very, very broad topic. Among them, what are the subtopics? The plant extracts can be used as a biopesticide. Aza directa indica is the best example. Then different hormones are used to prepare a trap where the insects are trapped and they are killed. Then the guppy fishes, they are also used as a biopesticides. In the water bodies, the fishes eat larvae and reduce the pest. In addition to that, different fungi, different bacteria, and different viruses are also used as a biopesticide. And among them, the topmost is Bacillus anthracis. They produce a delta protein, and nowadays genetically engineered plants are also available. This gene producing the protein, this protein which kills insect is produced by plant. That's why now genetically modified plants are also available. That is comes under the biopesticide. Then many fungi are there. The I think Bevaria is a common fungi which is commonly used as a biopesticide. Then insects are also used. Meaning is that insect kills insect. For example, trichogramma. Trichogramma is a one insect which kills other insect. Trichocards are also available in Bangalore. That also comes under the biopesticide. Then fungi, trichoderma is the very popular throughout the India. It is always said that only one fungus can give you crores of rupees if you produce on a large scale for a bio startup. So that is the outline I have given. And viruses are there. NPV, nuclear polyhedrosis virus, is a very common. They can kill the bollworm. So like that, I have given only the different types of biopesticide. Now I will go to the Bipran sir, and Bipran sir can proceed and give chance to everyone to explain their work. Thank you.
Thank you so much, sir. So once again, I welcome to everyone present here for this roundtable discussion on the topic biopesticides. So now I request each and every participant to please unmute yourself and if you have, you may share your research experience, your means, and if you want to put any comment or you want to share your knowledge. Now I welcome everyone here. So I request everyone to just one by one, you can unmute yourself. If it is possible, you can open your camera and please share your research experience. Dr. Prasoon, sir.
Yes, one by one,
Hmm.
Dr. Prasoon.
Yes, sir. Thank you. Thank you, Vipran, sir.
Thank
Uh,
you.
the, yes, sir. Just one new thing that I have come across regarding biopesticide is the utilization of lignin. Lignin can be valorized or repurposed as a biopesticide. So this is one kind of new arena that people are exploring while working at 2G ethanol plant at Indian Oil Corporation, Faridabad. I got to know that lignin residue comes out as a means in bulk after getting utilized all the cellulose and heavy cellulosic component. Lignin remains as such, which can be either be burned or can be repurposed. So in that, I hope I am audible, sir.
Yes, you are audible, Dr. Prasun.
Okay. So during that work only, the other research group, I was working on fungal enzymes. Uh, so on-site fungal enzymes I was working. So there another group started working on lignin. So they can take out vanillin from lignin, and the residual after vanillin extraction can be used as biopesticide. So that work was quite new, and I hope that maybe some of you are aware. But this is a new thing that I learned recently.
Thank you,
And since
Prasun, sir.
yes, yes, and since lignin, if TG plants are more because all the three prime oil producing companies of PSU companies in India, they all are focusing more on 2G plants. So more and more lignin are available and will be available in the moments for the valorization purposes. So maybe other uses of lignin can also be discussed, but from biopesticide perspective, I think this is a new aspect that raises new hope in biotech field. So thank you, sir.
Thank you, Prasun. So, dear students, if you have any means question to Prasun, sir, you may ask now. Usha Payal Maiti. Okay. Dr. Malini, madam, would you like to share your experience with us? Malini, madam.
Yeah, unfortunately, I haven't worked in this field. So, I mean, I joined to get some overview of from the participants or you are as you are conducting, because I have a plan for the students for doing some internship thing and likely that way. So, I'm not a speaker in this forum. I am a listener. Okay.
Okay, thank you, madam. Then everyone is contributed, madam.
Can I talk now?
Sure, madam. Yes, madam.
Yeah. So I am a senior principal scientist and I'm head of fermentation and microbial biotechnology division in CSI Triple IM Jammu. We have been working in this area for quite some time and we have the technology which can be used for biocontrol. So, trichoderma we have been using for long. We have processes and many industries have actually commercialized the technology. We have actually a fermentation facility up to 5 KL, and if any of the startups or any of the researchers who are working in the area of scale up for or fermentation, they can approach us, and we usually help the industries as well as the academicians, researchers for scale up work. And personally I have worked in trichoderma. So basically the biocontrol activity of trichoderma is usually due to the kind of peptides and peptidols that they produce, which have biocontrol activity. And currently I am working for biopesticide formulation for using the streptomyces species. So at this point of time I have funding for this particular project, and we have scaled up the process up to 500 liters, and we are going to patent the formulation, and if any industry can we can approach, we will be happy to transfer this technology. So thank you all, and I would like to invite people to collaborate with us. We will be happy to help them, whosoever would like to have the scale up work if they have optimized their work at lab level. So thank you so much for giving me the opportunity. Thank you.
Thank you so much, madam, for sharing your research insights.
Thank you.
Yes,
Dr. Asha, you will make for probiotics also?
Sir, probiotics,
Production?
I don't work, but we have worked with industries who have worked in this probiotic area. So if anyone wants, we have experience working in them, but
Yes.
for R&D, I don't work, but we
No,
can
not
help.
as R&D, for fermentation, basically.
Yes, we
Because-
do that. We do that. We have done already with one of the
Wonderful.
industries. Yeah.
Yeah, because one of our client wants to have a scale-up studies.
Yeah, sir.
So,
Sir, you may get them connected with
that
us.
is,
We'll be happy to help them. Yes, sir.
exactly.
Thank you so much, sir. Thank you.
Trichoderma you grow in a solid state fermentation or what?
Submerged
How you grow the
fermentation.
trichoderma?
Submerged fermentation.
Submerged.
Yes.
So you have the fermentation facility for solid state as well as the submerged fermentation also.
So
As well as
solid
the
state,
liquid broth also, right?
so submerged fermentation we have up to 5 kl, but solid state we are going to start soon.
Mhm.
So we have already procuring the bioreactor for solid state. We haven't started solid state yet, but within few months we will be starting that because our equipment is under procurement.
Wonderful.
Yes. Thank you.
Thank you, madam. So now, Dr. Halami Sana, we would like to hear from you, sir.
Wonderful. Thank you very much, Dr. Tiwari. As I have to say something. I will start first of all with my involvement, especially in the Department of Food Protection and Infestation Control, where we use the pesticide, the conventional pesticide, like we use for fumigation, uh, or infestation of disinfestation of the store grain pest. Okay. So why I am talking is because we use mostly the chemical pesticide, and when we use the like a chemical pesticide, they are persistent long term the effect which remains in the soil or in the environment. So this is one of this, and then of course they are the broad spectrum. They kills affects all the insects and even can affect the human being also. So this is how actually the biopesticides these days has a lot of importance from this perspective. Biopesticide, as we know, that they are less toxic than the conventional pesticides, and that's why we actually look more environmental friendly. They target only the pests which are closely related to them, not a broad spectrum like the conventional pesticide. Okay. Phosphine is a common fumigant, which we use especially in the store grain pest. And what happened in the recent past, methyl bromide and phosphine, especially the methyl bromide actually causes the problem of depletion of this ozone layer. With relation to the phosphine fumigants or phosphine as a pesticide has been shown to induce the resistance. So now most of this pest will get resistance also. The current aspect of the biopesticide is they work in a very small quantities and decompose quickly. So that is what actually more important is. So less exposure to the environment. And then because of all these attributes which I told, with the less toxic, affect the closely related organisms, and then they decompose quickly in the environment. They can be a part of what we call the integrated pest management.
Your voice is breaking, sir.
Can you hear me now?
Yeah, we can.
Yeah, so I was actually referring with reference to the biopesticide advantage over conventional pesticide. Before that, I told that what are the disadvantages of the conventional pesticide, especially giving the example of phosphine. And phosphine is used as a fumigant, especially in the store grain pest. And one of the major problem which we have reported in the phosphine is the development of the resistance on the phosphine also. Now, with reference to the biopesticide and from the microbial from the MBSI perspective, I would say that the Bacillus thuringiensis, the soil bacterium, which produces a Bt toxin, a crystal toxin, and affects the Lepidopterus insect. So it is actually more important, right? And it actually affects the gut, of this Lepidopterus insect, and this is how actually you can use the biopesticide or the Bt toxin, and then take care of this Lepidopterus insect, especially the caterpillar again, because the caterpillar are the one which actually they eat away the leaves, So if your economic part of the plant is the leaves, then you will have a heavy damage, or loss of too much loss in your agriculture field. So to develop a good biopesticide or genetically engineered, since it is a crystal protein or the protein base or the gene encoded protein, it is possible, what Dr. Deshmukh was telling, the plants producing a biopesticide, Bt toxin is possible. Why? Because these are the gene encoded proteins or gene proteins, So you have the corresponding gene. Those genes you can clone, put it in any plants or animals or microorganisms, so that microorganism or the plant will be producing this Bt toxin. And this is how actually you can develop even the plants resistance to the insect, right? What we call the transgenic plants, or the transgenic insects using this. So now we have seen some work we did, especially from the bioinformatics perspective. We have seen the large diversity of this crystal protein, endotoxin. What happens is actually there are actually the different components or the very high diversity of this Bt toxin has been reported, or the protein, which has been grouped in the several subgroups, and identified how effective or what are the domains actually are required for its activity. And also we did some work with relation to the bioinformatics perspective, and then we could actually see, because having the complete genome sequence of Bacillus thuringiensis is not difficult. Having this particular protein sequences is not difficult through the bioinformatics. Looking at the gene encoding those proteins is not difficult. So you can actually develop such a novel chimeric molecules also, which can be actually more effective and not found in the environment. So I will go on talking like this way only. Perhaps better we take some questions or the opinion of other participant also on this aspect. Okay. Thank you, Tiwari ji, for giving me this opportunity.
Thank you, sir, for sharing your knowledge. So, dear students, if you have any question or any participants, you have any question to the Usha Madam and the Prakash Salami, sir.
Yeah, exactly. Yes,
Just,
definitely.
yeah, you can unmute yourself, or you can just put in the chat box. I can read out from there. I think that one research scholar working on the probiotics. You may also ask any question if you have on the probiotics.
Yes, exactly. They also can be not a biopesticide, but they can be a biopreservatives, we can say.
Yes.
I believe one of the student is working on
Probiotics.
probiotics, so I can also provide some information.
So, Nabeen, Mineral Mathingis Kher. I think so. One question not related to this topic from a student, sir. Hello, am I audible to you, sir?
Yes, yes.
Not related to these topics. It's like means for its careers. Sir, I want to ask if I don't want to go in any research field. So, what other field is there with the good scope? Currently, I'm in the second year BSc microbiology, so I am not having more knowledge related to this field. Amna Hadri.
Yeah, yeah, you can go to the pharma industries or the food companies or the academic or the laboratory assistant position. Now several marketing, right? There are several such companies which actually offer the job in the pharma companies. Then industries, in the dairy industry, you need a technician, technical officer, technical person. You need in the pharma, the quality control officer or quality control assistant. Or then you, after the even BSc microbiology, can be a laboratory assistant for conducting a microbiology practical.
So, Amna Hadri, do you have any more question? Okay, sir.
Sir, there is one more question in the chat box.
Okay, sir. Sure, I will. So, one more from the Kher Swapnali. Due to the continuous research and use of these biopesticides can affect the biodiversity. Due to the continuous research and use of these biopesticides can affect the biodiversity.
Yes, definitely. That is what actually I was trying to tell. Since they have the broad spectrum, they will be killing the most of this now flora and fauna, the conventional pesticides. You know, they will be killing not only the useful insect, not only the crop damaging insect, but then they will be actually infecting even the useful insect, the insect which are associated with the pollination, butterflies, right? So such a we will be losing, in other words, the diversity. But when you use the biopesticide, since they are narrow spectrum of activity, they will be actually more selective. They will be inhibiting only the target pest, in other words. And this is what actually I told initially with reference to the biopesticide and then conventional pesticide. Then I told how the pesticide excessive usage or routine usage is became now a reason of the resistance development. You know, one of this participant was telling about the antimicrobial resistance from all the way from Nepal, Tribhuvan University. So that problem actually, yeah, that problem is there even in the pesticide also, in the regular usage of this conventional pesticide. Yeah. Similar to AMR.
Yes, sir.
And then there are such actually the insects which have been found develop resistance has been identified, and now very difficult to control those. And then there are totally new guidelines for this kind of studies. Exactly.
Prakash sir.
Yes.
Yeah. Sir, actually, the question I think was towards the research and usage of biopesticide, if they can also have the similar fate, like affecting the biodiversity. Biopesticides can do that, or it will not hamper any because they have narrow range or narrow spectrum. They will not disturb the biodiversity much.
Exactly. Yes. So now you just convert it another way. I understand.
Okay.
And that is what actually I also felt that telling what the major difference with reference to their spectrum activity and then usage, and then their impact on the environment. I thought that these are the more important points to be considered, when we are talking about the biopesticide. Though they will not kill your most of this insect sometimes, but then they are still effective. Why? Are useful? Why? Because of the environment concern. Why? Because they don't introduce the resistance which otherwise we see, and then they don't cause the damage to the environment or the even the insecticidal flora because of the reason of their the narrow spectrum, right? And then we need some insect. It is not that we have to kill everything. We need some of this insect for one or other purpose. I hope it is quite clear.
Yes, sir. Yes, sir. And to you, sir, means I have many things to learn from you. I always look up to this roundtable discussion to just to listen you because you have worked a lot and especially the. So what my question is now related to some kind of startup. If uh, we want students to engage in some kind of startup ideas related to probiotics.
Exactly. Okay.
So how can we initiate? So is it just a common lactobacillus that we can start with, or bifidobacterium culture, or we should isolate a new one, but it will take a lot of research, no? So
Yeah, but the best way is actually purchase the existing technologies where it is available, where you will get the culture with the scientific information of that and how the product to be developed. Okay, we have now the carrot nectar, just a 1 lakh rupees, 1,10,000 something. Lactobacillus plantarum is incorporated. And this culture actually identified with reference to the anti-obesity properties, gut microbiota modulation, reducing the cholesterol. And then when we actually use this carrot nectar as a substrate, so it takes care of the beta carotene requirement. And the vitamin A deficiency is very common in developing countries. Or otherwise, you go for the bifidobacteria. For that also, we have these technologies. Or otherwise, you don't want from us. Then you have to isolate the culture, identify, study, publish those work, and then see how you can actually, in what form do you want to give those product, right? So that is the another challenge.
yeah, yes, sir. That's why
Exactly.
because nowadays what we are seeing from students now, some of the means
Who were asked for research purpose, I am just giving what are the whether bifidobacteria or lactobacillus plantarum, or lactobacillus fermentum, or bacillus licheniformis culture. I am just giving. I don't have any inhibition or anything. You can even check against some of this insect also. Those who works with the insect, you know, but are fungi perhaps. Yes, possible. But then you just no, you can't aim at a time many targets, right? We can just take one target, see whether you look for a gut microbiota modulation, whether you look only for the a constipation problem or digestion, right? Or you look for the immunomodulation. So we have such already the identified probiotics. Incorporate, develop some food product, some juice, beverage, or the now buttermilk. We have this mission going on. But what is the culture we are using? Right now, we are not using any defined culture, right? Or we are using only the starter culture. We are not using a probiotic culture, right?
yes,
Now MBSI has made a buttermilk such a popular, but then
yes, yes. I am
no,
also involved in that 70 plus
exactly.
taking care of
Yes.
that. Yes, sir.
So in such cases, looking, using some probiotic culture, developing that, making the count with suitable, whether that required count of the probiotic culture is present or not in the finished product, and then market it. Now, most of this dairy, I think 30, 40 years back, no one was knowing dairy industry will be such a successful in India. Now you see, you will get tea, milk anywhere, curd everywhere, buttermilk everywhere, right? So like this way, I think one find a probiotics also will be like this. You go to Europe or US, everywhere, with the supermarket, you will have the 50% of this eating products, the probiotic product, the different type of the cheeses, the beverages. Just I brought from US, kefir, you know, almost some 8 probiotic bacteria are there. Some 4 lactobacilli and 4 bifidobacteria. I just got the 2 bottles from there. I am now trying to what I can say regenerate those and then see whether I can get the similar product. As such, we have also the several such probiotic bacteria and all. Yeah. So like this kind of innovative idea is required, which needs a really hard work, and then you see how best you can actually utilize. Yes.
Yes, because
Somebody wants to ask somebody. Yes.
so, sorry, sir. I was telling that so we can get this because some of the students are now more interested in initiating their own company or like a new product.
Definitely, definitely. You have. I hope you have my address,
Yes,
contact
yes,
details.
sir. I will email you once.
Just message me immediately. You will get the response from me.
Okay, thank you so much, sir.
Don't ask any financial assistance to start the startup. Huh?
No,
Knowledge
sir.
sharing
Because
is a free.
they are from good family, business family.
No, no,
They just
no,
want
no.
that okay something like
Just I am joking.
no
No,
problem,
no.
sir. Yes,
Knowledge sharing, the experimental part is sharing is not a problem. Technology, it is a safe property. Okay.
yes, sir. Obviously, because it's
Using the
after all,
culture for R&D, I will take care to help. But if you are using the culture for industry,
commercial activity.
for commercial purpose, then you have to pay not me, to CSIR, you
Yes,
know,
sir.
to CFTRI. Because they have to also earn no, something.
Ji.
Exactly.
Thank you so much, sir.
Thank you very much.
One question I have to Asha, ma'am. Sorry, because sir, yes, continue.
Hello, Asha madam is here.
Yes, please.
Please, ma'am.
Ma'am, I forgot to ask this trichoderma. What is the compound you mentioned that works as a biopesticide?
So small peptides and peptidoboles they produce most of the trichoderma strains, which are actually have biocontrol activity. So they are bioactive compounds. So some of them may have the potential for as antifungal or antibacterial activity. So they may act as the biocontrol agent, but there are some of the peptides which have pharmaceutical applications as well. So we have purified them and we have seen that some of have biocontrol activity and some of them have antibacterial activity as well.
Okay, so a small peptide means their molecular weight is less than some 30 kilodalton or even less than that. What?
So these are so they below 30 kDa, below 30 kDa.
Okay.
Yeah.
Thank you so much. And ma'am, also I will also contact you because for my polyhydroxyalconate, I may need some scale-up studies.
Yeah, but
Once I
sure.
finalize the data here, I will get in touch with you
Yeah,
sometime.
sure. So I'm in the group. You may message me on my number.
Sure, ma'am. Sure.
Yeah. Thank you. Okay.
Thank you so much.
Thank you so much, madam. So I have just shared one feedback form in the chat box. So please fill out the feedback form. It will help us to conduct the next session. So thank you all for your active participation and valuable insight in the today's roundtable discussion on the topic biopesticides. And our expertise, Dr. Asha madam, Anjana madam, Dr. Prakash
Yes.
Salami sir.
Can I have one question, please?
Sure, madam.
Can I ask? Dr. Asha, can you just tell me that which process you did for the that molecule, biomolecule?
So like when we ferment the microorganism at
Mhm.
large scale, then
Yes.
when we do the downstream processing, we make the solvent extracts, and
Mhm.
then through column chromatography we purify the peptides and
Okay.
characterize them. So characterization is done by multi-top MSMS. So
Mhm.
that's how we get their identification, and then with the amino acid sequence we completely get the sequence of the peptide that we produce.
Okay. Thank you so much.
Yeah, thank you.
Thank you, madam. So once again, thank you all for joining today's roundtable discussion. So thank you all.
Thank you. Thank you very much.
Thank you, sir.
Yes, sir. Thank you, sir. Thank you so much.